and my answer is going to be simple. We are nowhere with the malaria vaccine. The only one which is near licensed by WHO, at best protect 30% people of the people over a 6 months period. And so it's not a good vaccine, it's a very imperfect vaccine. And we still have to go to the drawing board and design new vaccine. We are not lucky as for COVID to have a good and efficient vaccine. All of them have their, if you want to design a vaccine which is specific, all of them have their characteristics. Intuitively, you would say that blocking the sporozoite to go into the liver would be the easiest one because the number of parasites injected is small, but the parasite is smart and has developed so many escape mechanisms that even this neutralizing a small number of sporozoite, we are not able to do it so far. So I would say if I was targeting one, I will try to target the liver again because you have small number of parasite. You don't have variation here that much compared to the blood stage or the sporozoite, but you need to induce T cells, and these T cells need to go to the liver. And again, the parasites in some ways avoid T cells. So we need to do more studies to understand the biology of the parasite and to really find the Achilles heel to target them. But we're still far from it. So the first bottleneck, I would say, is that we don't have what we call correlates of protection. Which mechanism we should target to induce a protective immunity? So we don't have that. We don't know if it's antibody level. We don't know if it's a T cell and what T cells should be induced. We don't have this qualitative measure of immunity. That's one. Second, we don't know which antigen or antigens to target. The parasite has more than 5,000 genes, and from these, some are expressed differentially at different stage, and we don't know which one to target and which one is associated with, again, with protection. If we knew which mechanism is involved in protection, which antigen to target, that will be a good start to develop a malaria vaccine. I think the new platform based on mRNA vaccines are quite interesting. They have never been tested in malaria so far, at least in a clinical trial in human. I know that they are being developed now for at least preclinical level. So let's see what it will be. But all the other platforms which are tested now for COVID and for other diseases have been tested in malaria and have not given very good results. Could be adenovirus, the DNA vaccines, other viral vectors, and none of them have been shown efficacy so far. And even prime boost combination, when you use one platform for priming and another platform for boosting, none of them are really demonstrated strong efficacy. So mRNA platforms are new. Let's see if they can perform better. There's still a lot to know and a lot to discover. So as I was mentioning, we don't know the exact mechanism. We don't know the exact antigen to target. So a lot for the newcomers to do and to find their niche in the future. So it's not like a small virus of 7 to 15 genes. We have 5,000, a very complex parasite, a very, I would say, interesting and challenging parasite. I think if you're interested in difficult problems and find new interesting solution, I will go in malaria more than I will go in virology, even if I like working in virology too. It will be a possibility if we knew which antigen to target, and if this antigen was conserved between species, that would be something we will be able to do. But even inside the same species, there are so many strains, and it's already a challenge to have a vaccine against so many strains. So the next step to go against multiple species will be another level of challenge. If I knew, I will get the Nobel Prize. So which is still open, as you were mentioning, for people wanting to go in malaria. There is a spot which is there for in the Nobel Prize for people who will discover a malaria vaccine. So if you want to be famous, go and find the malaria vaccine.